Good afternoon, students. In the last class, we studied about the amino acids. That means proteins. So proteins means they are the polymers of amino acids, especially alpha amino acids. Okay. So in that amino acids, they are classified into different types based on different categories. Okay. So if based on NH2 group and COOH group, they are classified as acidic amino acids, basic amino acids, and neutral amino acids. The thing here it is acidic amino acids means COH groups are more than NH2 groups. Basic means NH2 groups are more than COH groups. Neutral means both NH2 as well as COH groups are equal in number. Okay, and next one is essential and non-essential amino acids. Essential means which we require the amino acids that are useful for us, but we have to get them from the diet. They are not synthesized in our body. We have to get those amino acids or those proteins from diet. Okay. Non-essential means we need not to take the proteins by external force. They are automatically synthesized in our body. So those are non-essential amino acids. Okay. All are useful for human beings. Essential means we have to take that proteins by external source through diet. Okay, non-essential means they are synthesized in body. So in that, students, I explained that amino acid structure is R, C, NH2, here means H, COOH. This is the alpha amino acid. Okay, because this is the functional group means to this is the alpha carbon, to this carbon NH2 group is attached. As I told you, as the R group changes, then the name of the amino acid changes. Okay, I'll write some of the amino acid structures here. So if R group is H, then this becomes H, CH, NH2, COOH. Completely, if you have to write this becomes NH2, CH2, COOH. Correct? Okay. So this is a simple amino acid called as glycine. So students here, the amino acid is also equal to 1, COOH is also equal to 1. Equal number of amino and COOH group means this is a neutral amino acid. Understand? Next, if R is equal to CH3, then here in R group you have to write CH3, CH, NH2, COOH. So this one is alanine. Here also NH2 and CH2OH group are equal in number. So this is also a neutral amino acid. Okay. So next one is R group is CH, CH2, sorry, CH3 twice. R group is CH, CH3 twice. Okay. Next here, everything remains same. Here it is CH, CH3 twice. Only you are replacing what? R group. If R group is this, then this is valine. Okay, it is also a neutral amino acid because equal number of COOH and NH2 group is there. Okay, next one is leucine. That is equal to CH2, CH, CH3 twice. Same students here, extra CH2 group is added here. This one is, understand? Same group to this extra CH2 is added. So if this type of structure is there, so then our group becomes, this is the CH, COOH, NH2, here it is attached, like CH2, C, CH3 twice, This is the structure. Understand? Just I am replacing R. So this one is leucine. Okay, this is also a neutral amino acid. Okay, next one is the R group is iso. I will consider students. So listen, here one carbon is there, CH, CH2, CH2, CH3, CH3. For one carbon atom, so here two carbon atoms are attached. That is iso. Understand? This is the R group. If this is the R group, then this becomes CH, COOH, NH2 is the this group. Okay, R group is replaced by this. That is CH2, CH, CH3, CH3. Here it is isoleucine. Why? Because both the compounds are same. Molecular formula is same. This is also leucine, this is also leucine. Why it is called as iso? Because here to the second carbon atom the CH3 is attached. Understand? So this one is leucine, this one is isoleucine, this is also a neutral amino acid. Understand? So these five, which one is glycine, alanine, valine, leucine, isoleucine. So students, they are represented by, this one is by capital G, capital A, capital B, capital L, and this one is capital I. This is the representation of nucleic acids. Means in the DNA and RNA, coding and decoding is there in DNA. For example, if G is there, A is there, B is there, L is there, they are coded by another, like uracil, and here thymine is there, I think. Okay, so they are going to, these amino acids are present in nucleotides and nucleosides which are present in DNA and RNA, they involved in coding. So for that coding we require these letters. Understand? So this one is glycine, alanine, valine, leucine, isoleucine. Understand? So these are represented by single word as well as three-letter word. Means glycine means it may be written as gly, ala, val, leu, and this isoleucine means ILE. Starting three letters also represented by respective, so represent the respective amino acid. If they mention G, then also it is glycine. If they mention GLY, then also it is glycine. They may be represented by either three letters or a single letter. Understand? This is all about a neutral amino acids. Next we will discuss acidic. For acidic, our group is COOH, CH2, CH2. This one is what? R group. See, if this is the R group, then the amino acid, first you write this one, CH, COOH, NH2. Correct? And this is the R group. CH2, CH2, COOH. Correct? So here, if you compare this student, here only one amino group is there, two carboxylic acid group is there. If the number of carboxylic acid group is more, then it is acidic amino acid. Okay, name of this acid is glutamic acid. is acidic amino acid. Understand? Next one is our group is COOH, single bond CH2. This one COOH, CH2. So here our group represents same first. So this one is COOH, NH2. This is common. And to this CH2 and COOH group is attached. So this one is aspartic acid. This one is aspartic acid. So that means students, this is also an acidic amino acid. Why? Because here number of COH group is more than the amino group. Okay. So next one is our group is NH2, C double bond O, CH2, CH2. If this is the R group, then the amino acid becomes. So first we have to write CHCOOH, NH2, and this one CH2, CH2, C double bond O, NH2. So students, if you compare here, two amine groups are there. One COOH group is there. If amine groups is more than COOH group, then they are basic amino acids. Okay, so listen, compare these two structures. If CH2, C double bond O is there, then this is called as glutamic acid because COOH is there. So here it is called as glutamine because here the functional group is amine. Here COOH is there, this R group is same, C double bond O is same. If OH is there, glutamic acid. If NH2 is there, that is glutamine. Understand? So that means here number of NH2 groups is more than COOH groups. Next another one is asparagine. This one only. Here R group is. So students, COOH, CH2 is there, correct? So here C double bond O, CH2, NH2. Okay, so here it will become CH, COOH, NH2. This one is CH2, C double bond O, NH2. So students, this is asparagine. This is also a basic amino acid. Remember, comparatively I am writing. This functional group is the CH2, then it is glutamic acid. If it is COOH, if it is NH2, glutamine. Same here, CH2 is there, C double bond OH is there, that is acid group, then it is aspartic acid. If NH2 is there, then it is asparagine. If you remember this one, that is it. Because for one marks there is like, what is acidic amino acid? Given example. What is basic amino acid? Given example. So another one is this one, that is heterocyclic amino acid. Heterocyclic means, cyclic means consists of a carbon and its compounds in a cyclic manner. Heterocyclic means at least one atom is different other than carbon. Okay, so to that only one example is there, that is histidine. And right here it is a different structure and very important, histidine. Okay, what is the structure of histidine? Is a 5-membered ring. Here are the carbon atoms. Here it is a nitrogen atom. Okay. Alternate bonds. Here it is CH2. This is the structure of histidine. This is a heterocyclic amino acid. Heterocyclic why? Because listen student, if all are like this, then it is a homocyclic. Why? All are made up of carbon atom only. But here At the place of two carbon of nitrogen atoms are there. Hetero means different, other than carbon. That's why this is called a heterocyclic amino acid. Understand? So this is all about amino acid structures and their classification. So students, the next one is in the structure in our body, how the proteins are going to form. As I told you, the amino acids are the polymers. Proteins are the polymers of amino acids. Understand what I said? Proteins are the polymers of amino acid. So in our body, only amino acids are present in the form of proteins. Alone, they are not like glycine, alanine, they are not in that form. They are in a polymer form, means in the form of a protein. Okay. So students, next one is the structure of protein. Okay. So in the structure of protein is listen. So initially I will write here. I take one example of R, CH, COOH, NH2. Okay, students, I just to replace this R and NH2 group. Here it is NH2, here it is R. Understand? Consider another molecule of amino acid. This one is R dash. Now consider another one. As I told you, this R dash, R double dash may be C group, H group. That depends. Now I represent the R groups now. So on the basis of that, we can select any one R group. Okay. So students here, well this alpha amino acids, those are all the monomers. They form polymers. These are all monomers in single units. They form polymers in so many number of units repeat. When they will form, listen, here I NH2 is written as NH and H. Correct? So here, from here, C double bond O is written as, COH is written as like this. Correct? So write like this. So students, by observing you can easily say there is a removal of water molecule. Correct? So then it forms NH2, CH or C double bond O. H2O removes things followed by NH, CH or COOH. Okay, so they will form this bond, C double bond O, NH. This is amide functional group. C double bond O NH is an amide functional group, and this bond is called as polypeptide bond. A polypeptide linkage. Sorry, not polypeptide. Peptide linkage or a peptide bond you will see. Okay, students. If the bond is formed in between two amino acids, then it is a dipeptide. If it is in between three, then it is tripeptide. Bond is only one, but it is in between how many amino acids? Two amino acids. So then it is a dipeptide bond, means the bond formed by two amino acids. Okay, if here also same text is like C double bond O, here NH, OH is there, H is there, water molecule will be. Here also continues like C double bond O, NH, CH, COOH or double dash. This one is R dash. So here also one peptide bond is there, here also one peptide bond is there. Here how many amino acids are involved? 3. Then it is a tripeptide bond. And a tripeptide linkage. Understand? So how many number of amino acids involved in the formation of the protein? So that much of peptide bond we are going to say. If 4 amino acids, 10 tetra. If 5 penta. If 6 hexapeptide. 7, 8, 9, 10 decapeptide like that. So more than 100. That's why they are called as polypeptide bonds. So the proteins are the polypeptide polymers of the alpha amino acids. Understand? The bond between them is a polypeptide bond. How it forms? By the removal of water molecule by COH group and NH group. So removal of water means condensation. So these are all the condensation polymers of the alpha amino acids. Understand? So this is the structure. So here in the protein, what is going to happen here, students? These bonds are going to arrange, or the alpha amino acids are arranged in a straight manner, or a series manner, in a regular arrangement, one by one by one. Correct? So students, in the structure of protein, again primary, secondary, tertiary, quaternary structure is. How? Listen. And write the structure. So first one is primary structure. Primary structure means I consider them as a spheres, amino acid as a spheres. If they are continuously, regularly arranged in a straight chain. Okay, so this is a primary structure of protein. Understand? Next, secondary structure is. Secondary structure means, so listen, here CO group is there, here NH group is there. So here also CO group is there, here also NH group is there. At the same time, there may be a possibility of presence of extra NH2 and extra COH group here. Correct? So then what happens? So students, if any one alkyl extra highly electronegative atom is there, hydrogen is there, then they form a hydrogen bond. Understand? So when they form hydrogen bond, students, they start bonded like this. This one bonded here, again this one bonded here, this one bonded here, like this. They started arranging. They started forming hydrogen bond with the highly electronegative atom. Then this arrangement is arranged in this manner. This is called as alpha helix structure. Understand? The straight chain, the primary structures are arranged in a different shapes. The primary structure changes their shapes due to what? Formation of hydrogen bond. Okay, so this is one type of structure called as alpha helix structure. Another one thing that they are arranged in the form of a sheets like this. Okay, they are arranged in the form of sheets called as beta pleated structure. Beta pleated structure. Alpha helix and beta pleated. They are arranged in the form of sheets, that is beta pleated. They are arranged in the form of this helix form. You know helix form of a DNA you studied, I think so. Helix model of DNA is in this form only. Okay, the alpha helix structure. When they form a hydrogen bond. Okay. So next student, once this structure happens, the secondary structure, again it changes to tertiary structure. What is that tertiary structure means? The folding of this type of structures also takes place, means second time folding. This one is first time folding. Here also folding takes place. Okay, next second time folding. So if the second time folding happens, students, they form this type of structures. Understand? Means this type of helix models here, they again fold with each other. So this one is what? Second line folding. This is a tertiary structure. Okay. Next, for quaternary structure, complete, and at this time they form fibrous as well as globular, fibrous as well as globular proteins. You know that fibrous protein, they are in the form of a fiber. Globular protein, they are in the form of a circular manner. Understand? So example for a fibrous protein is keratin. That is, as you know, hairs are in a straight form. It is like a thread form. Fibers form. Keratin, you know. Keratin present in what? Hair. Right? So and also it is present in muscles. In muscles means, you know, muscles are, they are here by layer, they arrange muscles. They are in a thread-like form. Example is keratin, and also another one is myosin. Okay, myosin which is present in muscle. This one is example is keratin and myosin. And students, these two are water insoluble. Understand? Fibrous proteins are water insoluble, which is important for one. What are fibrous proteins? And give an example. Fibrous proteins means the tertiary structure of alpha amino acids are a tertiary structure of proteins which are in the form of a fiber. Example is keratin and myosin. So they are water insoluble. Means if you take an air, if you drop in water, means it never dissolves in water. Understand? Next. Globular protein means they are arranged in the form of a circular manner. They form a globe-like structure. Understand? So these are water soluble. These are water soluble. Example is insulin. You know, students, the diabetes patients are there. So they often used to go for urination, right? Because they experiences lack of production of insulin. If they intake insulin, it is a water soluble. Understand? So the urination formation takes place in a very fast manner in diabetic patients rather than normal human beings. I think you observe that. Understand? So insulin, another one is albumin. These two are the globular proteins and these are soluble in water. This is also important for one. What are globular proteins? And example, insulin and albumin. Fiber means fiber like. Example is hair. Keratin. Okay? Don't write hair, it is keratin. Next, for globular protein, it is insulin. This is what? The tertiary structure. The last one is that is quaternary structure means they are completely folded like this. They form a complex structure. Third type folding takes place, and then these type of structures again folded. So they form this type of structures. So so this is all about structure of protein. Primary, that is only the arrangement of alpha amino acids. Secondary tells about shape. Tertiary tells about the function. And last one is it is actually the structure of the protein. Understand, students? And the next one is denaturation of protein. Denaturation means breaking of the bonds. Here polypeptide bond is there, correct? So breaking of that bond is called as denaturation of protein. Understand? The best example is boiling egg. You know, if you boil egg, then the whatever the liquid is present inside the egg, yolk is there and white color, that is albumin is there, correct? Your egg yolk, yellow color part is there, white color part is inside the egg. So if we boil that, that will solidifies. Once it solidifies, we cannot change its state. Understand? Once the egg boiled, once it converted completely into solid state, solidifies, we cannot change its state. Understood? Are you getting my point? So, what happens there? Actually, the structure in the egg is in this form, quaternary form. When we supply it, the bond breaks and converts to tertiary. Tertiary converts to secondary. Secondary converts to primary, and primary is the final structure. Understand? So, in the denature of proteins, only the secondary and tertiary and quaternary bonds will break. The primary structures remains in the protein as it is. The structure of the primary structure never breakdown never takes place. Only the secondary, tertiary, and primary quaternary structures breakdown takes place. They keep on unfolding. Understand? If primary folds, secondary will form. Secondary structure folds, tertiary will form. Tertiary will fold, quaternary will form. So similarly, while we are during denaturation, quaternary will unfold. Unfold means separate. Then tertiary will form. If tertiary unfold, secondary will form. Secondary unfold means primary will form. That is called denaturation of protein. Best example is boiling of egg. Understand? And this process is not reversible. Once it is done, then it is done. We cannot reverse it. Understand? So this is all about the proteins. So remember students, you have to remember in the structure of protein means the structure of protein it is explained by the formation of the peptide. If the peptide bond is in between the two amino acids, then it is called as dipeptide. Three amino acids means tripeptide. Similarly, based on amino acid, they are classified as polypeptide bonds. Okay, so this is what the primary structure. Arrangement of alpha amino acids in a regular manner or a regular pattern. Okay, if the hydrogen bond takes place in the primary structure, they get some shape. That is secondary structure. One is alpha helix, another one is beta helix. If these secondary structures folded, then they form tertiary structures. That is 5% globular. Fibrous proteins or fibrous structure means it is the arrangement of the tertiary structure of the alpha amino acid, which is in the form of the fiber. Okay, and it is water insoluble. Next, globular means it is also a tertiary structure of the alpha amino acid, but it is present. Sorry, the shape of that is in a globule form. Understand? So here they are water soluble. Here example is insulin. Here example is keratin and myosin. Keratin present in air, myosin present in muscles, and these are inside the body. Okay, they are soluble in water. You know that insulin means a particular gland secretes the insulin hormone. Correct? Next, another one is that is quaternary structure. When tertiary structures folded with each other, then the quaternary structure will form. To remove that all the structures from quaternary to primary is called as denaturation of protein. Understand? So this is a very important part of the biomolecules. Understand, students? So the next one is vitamins. Vitamins. Next one is vitamins. So listen, vitamins means you may say it as vitamins or vitamins. Okay. So those are all organic compounds which we require very less quantity for our body, but they are very essential for the normal growth of the body. Understand? So these are the organic compounds we require very less quantity, but they are very essential for the growth, normal growth. Understand? For example, vitamin A is there, vitamin B, C, D, E, and K. These are all very important. Vitamin A is, if deficiency is also there, then also it affect. If excess is there, then also it affect. Understand? So they require a minimum quantity for the normal growth.